So we come to the next tryptophan, which is the repressible operon. As I told you, the operons are of two types. One was inducible and another was repressible. In inducible operon, we studied about the lac operon, which is there to digest lactose. And now we were doing tryptophan operon. As I told you, this operon is repressible, repressible operon. Repressible operon means this is mostly normally in switched on condition. Normally in on condition, but can be switched off. Normally it is in on condition, unlike the inducible, which is normally in off condition but can be switched on. This is normally in on condition. Normally it is in on condition, but if required, it can be switched off. Now I was telling you about the operon here. I told you that this the DNA segment is like this, in which we have this P promoter gene, repressible gene, again promoter gene, operator gene, and here we have 5 structural genes. This way, this was promoter, this was repressible, this was again promoter, then operator, then we have structural gene, and the name I kept was A, B, C, D, E. I told you the various names, and I told you that to the promoter, both the promoters, what is added? CAMP gets attached, and along with CAMP, one more thing which gets attached is RNA polymerase. RNA polymerase. This attaches to this. Okay. Same way here also CAMP and RNA polymerase attaches to this P also promoter. This attaches. Now R is the repressible gene, which is inhibitory gene. R is repressible or inhibitory gene, and because it is inhibitory, that means whatever protein is formed, it will inhibit the function. Now what happens that when this RNA polymerase is attached to this promoter, this slides over this. The RNA polymerase slides over this, and by the process of transcription, messenger RNA is formed, and this messenger RNA undergoes the process of translation, and as a result of translation, protein is synthesized. This protein is in inactive form. This is in inactive form. This protein is in inactive form. And because this is in inactive form, this protein, which is also known as repressor protein. So another name for this is repressor protein. And this repressor protein is inhibitory in action. Inhibitory in action. But this is inhibitory in action, but right now it is in inactive form. So because it is in inactive form, it cannot show its inhibitory function right now. So because this is in inactive form, it will not bind to the operator. It will remain like this. When it does not inactive form, when in inactive form, it does not bind to the O site or operator site. So when it does not bind over O site, what will happen? This RNA polymerase will slide over the structural gene. So when it does not bind, so RNA polymerase keeps on sliding over structural gene. It keeps on sliding over structural gene, and when this RNA polymerase slides over this structural gene, because nothing has bound to the O site, so no hindrance on the way, so this RNA polymerase will slide over this gene. As a result, what will happen? in all the structural genes, transcription will take place, and this transcription will result in the formation of messenger RNA, and this messenger RNA undergoes the process of translation, and as a result of translation, the protein is formed, and this protein here is tryptophan synthetase. Tryptophan synthetase. And this tryptophan synthetase, which is a protein which is formed, this is an enzyme protein. This is an enzyme protein which is formed. This enzyme protein, what it does that this converts the raw material. This converts the raw material in the presence of tryptophan synthetase. I am writing in short. This raw material in the presence of tryptophan synthetase gets converted into tryptophan. That means a lot of amino acid is available. That means this is normal condition, normal condition, normal condition. This is normal condition which is switched on state. Switched on state. This is the normal condition which is switched on state. So what happens that because of this switched on state, transcription, messenger RNA, translation, tryptophan synthetase will form, and whatever the raw material is there in the presence of this enzyme will get converted into tryptophan, and lot of tryptophan will be available all the time, which is because this is an essential amino acid and required, by the body. So all the time tryptophan is available to the body, because this is normal condition is switched on state, and this is switched on state. Switched on, see, everything is, the process is going on. This RNA is sliding over the R gene. This RNA polymerase is sliding over the structural gene. So this is switched on state because the continuously the process, transcription, translation, sliding over of RNA polymerase is taking place. So this is not in normal condition because this is repressible operon, this is on, and the complete synthesis of tryptophan synthetase takes place, which converts the raw material into tryptophan, and all the time this essential amino acid which is tryptophan is available to the body. Is it clear, children? Is it clear, or any query in this? Yes, any query, or is it clear? Yes, beta. Any query in this, or is it clear, this tryptophan? Okay, Sukhsahaj. Yes, others, just let me know, is it clear, or any query in this? Okay, Paras. Yes, others. Sukhsahaj, Paras, I've got your message. Others? Yes. Chitra, Vanshika, Simran, Aditi, Mitali, Girish. Okay, I've got your message. Yes. Mubashir, Navneet, fine. Mubashir, Aditi, Amit Katoj. Okay, yes. Tanish, fine. This is clear. So this is the normal condition. Like, if you remember lac operon normally was in off state. Lac operon, which we studied, lac operon, which was a inducible operon. Inducible operon. This was normally in switched off state. Normally in off state. This is normally in on state. This is normally in on state. But lac operon was normally in off state, and when outside the cell lactose was there, this lactose, when it entered, then only it attached to the repressor protein, and it didn't let it attach to this O, and no synthesis took place. So that means normally lac operon was in off state, but normally this repressible operon is in on state. Now what happens? Now what happens when this tryptophan operon, this repressible operon, is in off state, or how, suppose whatever amount of tryptophan is required by the body is synthesized, like say 100 units, 100 units of tryptophan is required by the body, and this 100 units has been synthesized. That means no more tryptophan is required now. Or suppose say 100 units is required, and the amount of tryptophan formed here is 100, say 10, 110 units has been formed. So this is more than what is required by the body. So this is extra, 10 is extra, because the body needs only 100 units of tryptophan. Now, because of the switched on state of this tryptophan operon, 110 units have been formed. That means extra has been formed. Now what happens? That means this operon must be switched off now, because no more tryptophan is required. So it should be switched off now. Now what needs to be done to switch off this operon? And how this operon will be switched off? This operon will be switched off when this repressor protein, which is in inactive form, it will be activated. So when this repressor protein will be activated, it will go and bind at the O site, and then this RNA polymerase will not be able to slide over the structural gene, and then no formation of tryptophan synthetase will take place. Could you understand? Could you understand? Okay, so I'll show you what happens in that case. Here, I'll just make it here only. Now, as I told you, suppose the tryptophan required by the body is tryptophan required by the body is 100 units, and the amount formed is 110 units. So that means 10 units extra has been formed. 10 units are extra. And these 10 extra units, 10 units are extra. So because extra has been formed, now it is not required. The body doesn't require any tryptophan. So what happens? See, here in any way sliding will take place, because this area up to this repressor gene, there is no operator. So this P, which has RNA polymerase will slide over R, transcription will take place, and as a result of transcription, messenger RNA will be formed. This messenger RNA has been formed. This messenger RNA will, by the process of translation, will form repressor protein, which is in inactive state. This is inactive repressor protein is formed. Now what happens to this inactive repressor protein? To this inactive repressor protein, these extra tryptophans are added, plus extra tryptophan. Extra tryptophans are added. When to the inactive repressor protein, extra tryptophans are added, the repressor protein becomes activated. So we get activated repressor protein. As a result of this, the repressor protein gets activated. That means it has become, now I'm making a particular shape of this. That means this repressor protein has become activated. When this repressor protein gets activated, what will it do? It will go and bind with at the O site. It will go and bind at the O site. This is O site, and it has gone and bind with the O site. It goes and binds with the O site. This is the O site where this repressor protein, now which is the activated repressor protein, it has gone and it binds with the O site. Now, because it has now bind to the O site, this RNA polymerase, which is at P site, because of this obstacle, it cannot slide over the structural gene A, B, C, D, E. Because of this activated repressor protein, which is present here, this RNA polymerase cannot slide over the structural gene. So when this RNA polymerase does not slide over the structural gene, that means no transcription, no transcription. no messenger RNA, no messenger RNA. And when no messenger RNA, no translation. When no translation, no tryptophan synthetase is formed. No tryptophan synthetase is formed. When no tryptophan synthetase is formed, then the raw material does not get converted into tryptophan. Does not get converted into tryptophan. So it does not get converted into tryptophan. That means no tryptophan is available. No tryptophan, because what the body required, that much tryptophan has been formed. So no this activated repressor protein, with the repressor protein which is activated now, it goes and binds with the O site, and because of that the RNA polymerase cannot slide over the structural gene. So no transcription, no translation, no synthesis of tryptophan synthetase, and no conversion, no formation of tryptophan. And then that means what happens normally on, now what is this condition? This operon, tryptophan operon in switched off state. So now it is in switched off state. So that was on. Normally it is on, but when body requires, when body says that I have sufficient tryptophan, so it can be switched off, and the synthesis of tryptophan can be stopped, okay, by activating the repressor protein. And how it gets activated? The repressor protein gets activated when the extra tryptophan adds to the inactive repressor protein. It gets converted into active repressor protein, which binds with the O site. And when it binds with the O site, the RNA polymerase cannot slide over the structural gene, and there is no formation of tryptophan synthetase. Is it clear to you, children? Is it clear to you? This tryptophan operon in switched off state. Yes, beta. Any query in this? Okay. Yes, beta. To everybody, if you want me to repeat this, I'll repeat this. Yes. Yes, others also. I want your response. We have done both the types of operons, the inducible operon, which is lac operon, and the repressible operon, which is tryptophan operon. So both the operons we have done. Although we have just one in your book, in the NCERT book, this is not there, but still I have done so that you need it for your exam. If you need it for your exam, it will be helpful. Yes, beta. Yes. Yes, I want your response. Any question regarding this? Any question? Yes, bache. Any question in this? Yes. Yes, Chitra, Samarth, Mariam, Vanithi, Jasmine, Karthik Thakur, Shruti Lakde, Gunjan, Amit, Arif, Girish, Mubashir, Parthavi, Sheetal, Sukhman, Sukhsahaj has answered me. Suzanne, Tanish. Yes, bache. Any question regarding this? Any query? Or anywhere you want me to repeat? Yes, bache. Okay, Shruti is saying no query. Others, Chitra, is it clear? Samarth? Samarth, are you understanding? Mariam? Yes, Mariam. Yes, beta. Okay. Mariam, you have a question or you understood? You are writing yes, ma'am. What does it mean? You have a question or you understood it? Yes, beta. Okay, you understood, Mariam. Fine. So I think everybody has understood it and we have just 1 minute left. So tomorrow we will start with the next topic. And right now I'll end my lecture. So keep safe. Okay. Okay, beta. Stay fit, stay healthy.